insightful look into cleaning validation, what it is, why it matters,
Yeah.
and how it all works in practice. So I guess our mission today is to kind of unpack this seemingly,
Got
technical area and show why it's actually so essential for patient safety
Yeah.
and product quality.
It's all about the patient at the end of the day, right?
Exactly. So we'll be drawing directly from Wyman's insights here
Okay, so
to kind of guide our discussion.
right.
So let's start with the basics. I guess, what is cleaning validation
So-
in simple terms, and why is it such a big deal in pharma manufacturing?
Yeah, so when we think about pharmaceuticals, right? The focus is often on the drug itself. The active
Yeah.
ingredient, the what's going to make people better.
Right.
But the journey a medicine takes the equipment it interacts with
Yeah.
during manufacturing, all of that has to be meticulously controlled.
Oh, yeah, for sure.
And this presentation really highlights that cleaning isn't just,
Uh-huh.
a routine step. It's a scientifically validated process.
So it's not just like, oh, wipe it down with some,
That's right.
soap and water and call it a day.
No. It's much more rigorous than that.
Okay, so then what does it actually mean to validate a cleaning process?
So basically, industry standards like the Q7A, Good Manufacturing Practice, they mandate that cleaning procedures are proven effective. And this isn't theoretical.
Right.
The validation has to reflect exactly how the equipment is used day to day. So the validation plan itself is incredibly detailed. It covers everything from the specific equipment, at the cleaning steps, the materials used, how samples are collected, what level of cleanliness is acceptable, and even the lab tests that are used to verify it.
It sounds super thorough. I mean, why all the fuss, I guess?
Well, think about it, contamination.
Okay.
If equipment isn't properly cleaned, residues from a previous product or cleaning agents or even environmental microbes,
Right.
they could end up in the next batch of medicine.
You don't want that.
No, you really don't. And for certain products, especially injectables,
Oh, yeah.
the presence of even tiny amounts of microbes or their byproducts, which are called endotoxins, can be extremely dangerous.
Oh, for sure.
So cleaning validation is really the bedrock of preventing this.
Okay.
It demonstrates with evidence that the cleaning process consistently removes these potential hazards.
So it's about having proof that it works every time.
Exactly. Every single time.
Yeah.
And this allows manufacturers to confidently use the same equipment for different medications
Okay.
without risking what we call cross-contamination.
Right. Makes sense.
And there's a PDA technical report number 29,
Okay.
which really emphasizes how critical this is within the industry.
So it's not just like one company's thing.
No, it's an industry standard.
This is like everybody's got to be doing this.
Absolutely, yeah.
Now, the presentation talks about a cleaning validation lifecycle.
Right.
What are the key stages there?
So the life cycle is a three-stage approach to ensure cleaning effectiveness from development
Okay.
all the way through ongoing use. So stage one involves these initial small-scale studies to really understand how different cleaning methods work with specific residues.
Oh, okay.
And for biotech products, this initial stage is particularly important
I can imagine, yeah.
because biological materials can be much more challenging to remove than traditional, chemical compounds.
Right. You think about like sticky proteins.
That's exactly. Yeah. Yeah, sticky proteins, things like that.
Yeah, that's not going to come off with just, some soap and water.
Exactly. So you got to figure out what works.
So what happens in stage 2 then?
Stage two is where you establish your acceptance criteria.
Okay.
So basically, what level of residue is considered clean enough and how you're going to measure it.
Right, so you're setting the bar.
You're setting the bar, exactly. And again, for biotech, this often requires very sensitive analytical methods
Oh, wow.
to detect even minute traces of biological materials.
So it's not just like looking at it and being like, yeah, that looks clean.
No, you need sophisticated equipment to make sure it's really clean.
Okay, cool. And the final stage, stage 3.
Stage 3 is all about continuous monitoring.
Okay.
So it involves the ongoing collection and statistical analysis of cleaning data
Right.
to identify any trends or potential issues over time.
So it's not a one and done, you're constantly checking.
Exactly. It's a life cycle you're constantly checking,
Got it.
making sure things are working the way they should be.
Now, the presentation mentions AstraZeneca's Biologics Frederick Manufacturing Center,
Yeah.
which actually won an award. How does that facility fit into all of this?
So that's a really good real-world example. They were recognized as ISPE's Facility of the Year in 2011.
Oh, wow.
And that suggests that they demonstrate best practices in all aspects of pharmaceutical manufacturing.
Okay.
And, robust cleaning validation programs would definitely be a key part of that.
So they're like the gold standard,
Yeah,
basically.
you could say that.
Yeah.
They're definitely a leader in the industry.
So it's not just, theoretical. This is something that's being put into practice today.
Exactly. It's happening right now in top facilities around the world.
At the highest level.
Yeah.
So I guess, to wrap things up, this deep dive into Wyman's presentation really reveals that cleaning validation is a non-negotiable, science-driven process with a life cycle approach. It's not just about appearances. It's about proving at every stage that the manufacturing equipment is safe for producing the medicines that,
It's absolutely we all rely on. It's a testament to the scientific rigor that underpins the entire pharmaceutical industry.
Yeah, it really is. It makes you think, considering the strict regulations and this long-term life cycle approach, what kind of hurdles do you imagine companies face
Oh, there are a ton.
in maintaining effective cleaning validation across all their different manufacturing setups in such a wide range of products?
Yeah, you've got different products, different equipment, different processes. It's a constant challenge.
It's got to be a logistical nightmare.
It can be, yeah, but it's absolutely essential.
When it comes to making medicines, cleaning is anything but simple.
Absolutely. We're talking about pharmaceutical manufacturing here. And the big question is, how do these companies ensure that their equipment is completely clean between batches of different drugs? Think of it like a super detailed, rigorous spring cleaning, but for making medicine.
It really is. We're diving into the life cycle of something called cleaning validation, which is a really systematic process that basically proves that manufacturers can reliably eliminate every single trace of the previous product from their equipment.
Because obviously that's like super important, right? You don't want any leftover residue contaminating the next batch of medication. It's all about making sure every single medication is pure and safe for the patients taking it. And the FDA has some pretty strict guidelines on how to do this right.
Right, and those guidelines outline 3 really crucial stages, and those are what we'll be focusing on today.
Okay, so let's start at the very beginning. The first stage is, what is it? Process design.
Exactly, process design.
So this is where they figure out the absolute best way to clean, right?
Yeah.
I'm guessing it's not just like, oh, just grab some soap and water and give it a scrub. There's got to be more science to it than that.
Way more science. This stage is all about really understanding what it takes to get that equipment absolutely spotless.
Spotless, yeah.
And a core idea here is something called TACT. It stands for time, action, concentration, and temperature.
Okay, time, action, concentration, and temperature, TACT. Got it.
So think of TCT like a recipe for perfect cleanliness.
Okay, I like that.
You need just the right combination of those 4 elements to get the best possible results. Time, how long the cleaning takes. Action, which could be the force of spray nozzles or even manual scrubbing.
Right.
Concentration, meaning how strong the cleaning agent is. And of course, temperature, because sometimes you need a bit of heat to help things along.
So it's not just one thing. It's a whole carefully considered formula. What other things go into designing this cleaning process?
Oh, tons of details. They look at the equipment itself, the materials it's made from, how well it drains, even doing tests to make sure cleaning sprays can reach every nook and cranny.
Interesting. So like if there are any parts that are hard to reach with the spray, I got to figure out a way to clean those too.
Exactly. Got to be thorough. And then they have to think about the products they're manufacturing, because the cleaning solutions they use have to be effective against the specific chemical and physical properties of the drug ingredients and any other materials involved.
Makes sense. Different drugs, different cleaning challenges.
Absolutely. And then there's this really crucial part. They need validated tests to confirm that absolutely no residue is left behind.
Okay, so that means it's not just about cleaning, it's about being able to prove that it's clean to a scientific standard.
Exactly. Proof is everything in this business.
Now, I did notice something interesting in these guidelines. It seems like some biotech companies use something called an inactivation approach. What's that all about?
That's actually really interesting. So for some biotech products, they can actually demonstrate that the active ingredient in the drug, the part that actually has the medicinal effect, becomes harmless, we say pharmacologically inactive, during the cleaning process.
Oh, so they're basically deactivating the drug itself during the cleaning process.
Exactly. And they can do this by, say, using really extreme pH levels, like very acidic or very basic solutions, or by using high temperatures. And if they can prove that the active ingredient is inactive and also removed to a safe level, it can sometimes mean they don't need those super specific product residue tests.
So it's like if you can guarantee it's harmless, even if there's a tiny bit left, you don't have to worry as much about measuring every last molecule.
Right. It highlights a key principle. Really understanding the molecule's behavior allows for smarter validation strategies.
That's fascinating. And I read that they actually designed these inactivation studies on a smaller scale first, kind of like a test run in the lab before doing it in the full-scale manufacturing setup.
Yeah, they'll try to perfectly mimic the full-scale cleaning conditions in the lab, and they'll often use what we call worst-case scenarios, basically pushing the limits to make sure that the inactivation is really robust. And they try to keep these lab studies very focused, sometimes even simplifying steps to concentrate the residue.
Simplifying to concentrate the residue so it's easier to see if it's really gone.
Exactly. And a really important part of this is showing that the inactivation isn't reversible. You don't want it to, reactivate over time.
Right, that would defeat the whole purpose.
Yeah.
Okay, so they've got these lab studies running. They're simulating the cleaning. They're inactivating the drug. How do they actually check if it worked? What kind of methods do they use to actually see if the inactivation was successful?
Good question. So they need ways to basically see if the active ingredient has been broken down. So one common technique is something called SDS page, which is kind of like a molecular sieve, if you will. It separates proteins by size, so you can see if the larger active protein molecules have been fragmented into smaller pieces.
So if it's working, you should see smaller bits of protein instead of that big intact molecule.
Right. And there are even more sensitive techniques like capillary electrophoresis, which offers even finer separation and sensitivity.
Okay, so they're really looking for evidence that the molecule is being broken down into smaller, inactive pieces.
Exactly. And another method they use is SE-HPLC, which is also size-based.
Okay, so lots of ways to verify that the inactivation is actually happening.
Exactly.
All right, so we've designed this incredibly detailed cleaning process in stage 1. We've got ways to check if it's working. What's next?
Next comes stage 2, which is called process performance qualification, or PPQ for short.
PPQ, process performance qualification.
Yeah.
Okay, what happens in PPQ?
So this is where we take that carefully designed cleaning process from stage 1 and we actually put it to the test, not in the lab, but in the real manufacturing environment, using the actual equipment at full scale.
So it's like a real-world test drive to make sure it actually works in practice.
Exactly. And the goal of PPQ is to confirm that the cleaning process can be consistently repeated batch after batch at full commercial scale.
So it's not just a one-off success. It's got to work reliably every single time.
Every single time. Consistency is key.
What are they looking at during PPQ? Like, what specific things are they checking?
They're assessing all sorts of things. They look at the biochemical properties of the product and any intermediate materials involved just to make sure that the cleaning process is effective at removing them. They're verifying that their acceptance criteria, which is basically the maximum allowable residue, is actually being met.
So they've got a really strict limit on how much residue is allowed, and they're making sure they stay below that limit.
Right. And they're confirming that those TACT parameters we talked about, the time, action, concentration, and temperature, are being consistently applied in practice. It's all about making sure everything is done exactly as it was designed.
And I think I remember reading that there's a pretty typical sequence for the actual cleaning process, isn't there?
Yeah, there's a general flow that a lot of companies follow. It usually starts with an initial rinse with water, just to get rid of the bulk of the material. Then they use the cleaning agents, followed by a final rinse, this time with super high purity water, and then finally they take samples.
So they're rinsing, cleaning, rinsing again, and then checking.
Exactly. And those samples are often taken by swabbing those worst-case locations we identified earlier, the ones that are hardest to clean.
Got it. So now the big question. How do they decide how much residue is acceptable? You mentioned acceptance criteria earlier, and the guidelines talked about a couple of ways to figure that out.
Yeah, there are a few main approaches. One is called the 0.1% criterion, which basically aims to reduce the level of the previous product by a factor of 1,000.
So like if you started with a gram of residue, you'd want to be down to a milligram after cleaning.
Something like that, yeah. But increasingly, they're moving towards something called health-based limits, which are a bit more scientifically rigorous.
How so?
Well, these limits are actually derived based on the potential toxicity of any remaining residue, considering factors like LD50 values, which tell you how much of a substance is lethal to half a population.
Oh, wow. So they're actually looking at how harmful even tiny amounts of residue could potentially be.
Right. They also consider ADE and PDE limits, which are related to how much of a substance you can be exposed to without any adverse effects. These health-based limits are more directly tied to patient safety, so they're definitely preferred these days.
Makes sense. Erring on the side of caution. And I think there was an example in the guidelines about using something called total organic carbon, or TOC, to determine cleaning limits. How does that work?
Right, so they might take a sample of the final rinse water or swab a surface and then measure the total amount of carbon present in that sample. And assuming that any remaining residue is uniformly distributed, they can actually calculate a parts per million limit based on the amount of TOC found, the area swabbed, and the volume of liquid used to collect the sample.
So it's like a way to get a general sense of how much organic material, which could include drug residue, is still present.
Exactly.
All right, so we've designed this meticulously detailed cleaning process. We've proven it works in the real world. What's the final step in this whole life cycle?
The last step is stage 3, which is continued process verification, or CPV.
CPV. Continued process verification. Okay, what's that all about?
The stage is all about making sure that the cleaning process continues to be effective, that it stays in control during routine, ongoing production. It's about continuously monitoring the process to catch any potential problems before they can actually impact the quality of the product.
So it's not just a one-time validation, it's an ongoing process of checks and balances.
Exactly. They're constantly making sure that the cleaning process remains reliable and effective because patient safety is paramount.
Wow. It's incredible the level of detail involved in something that, on the surface, seems as basic as cleaning.
It's really a testament to the commitment to quality and safety in the pharmaceutical industry.
Absolutely. This whole deep dive into cleaning validation has been fascinating. It really makes you think about all the other seemingly simple processes in complex industries that might have this kind of incredible rigor behind the scenes. We often take these things for granted, but there's a whole lot of science and effort going on behind the scenes to make sure everything is safe and effective. And permitted daily exposure, PDE.
PDE, yeah.
So this is all about like how much of a substance, especially when we're talking about medicines, how much can you be exposed to daily without it causing any problems?
Right. And it gets really interesting when we think about how different medications are made,
Yeah.
often in the same facilities, right? So we're going to look at how this PDE is calculated, making sure your meds are safe, even when it comes to like tiny trace amounts.
Exactly.
We've got a great guide for this, a 2014 guideline from the European Medicines Agency, the EMA.
Ah, the EMA.
We're going deep into the recipe that makes sure your medications are safe.
It's like a recipe, yeah.
Yeah.
We're going to break down each ingredient in the PDE formula and figure out why it matters.
I like that analogy.
So first off, we've got this formula.
Yeah.
What's the basic layout, the structure of this PDE calculation?
All right, so the EMA guideline sets us up with a pretty clear framework.
Oh, good.
The formula goes like this. PDE equals NOAL, that stands for no observed adverse effect level.
Okay.
Theta times a weight adjustment. And then we divide all of that by the product of 5 factors, F1, F2, F3, F4, and F5.
5 factors. Okay, that seems...
A lot to unpack.
Yeah, we got a lot to unpack there. So let's start with that first part. You mentioned NOAEL.
Right.
So what's that all about?
So NOAEL, the no observed adverse effect level, is basically the highest dose of a substance that doesn't cause any harmful effects in scientific studies.
So it's like the top limit.
Exactly. It's our starting point for determining what's safe.
Okay.
This data usually comes from rigorous toxicology studies, often using animal models.
Makes sense.
Right.
All right, so then we've got this NOAEL.
Yeah.
And then you mentioned multiplying it by a weight adjustment.
Right.
Why do we need to adjust for weight in this equation?
That's a great question.
Yeah.
So you see, the NOAEL we get, it's often derived from studies on animals or even adult humans. But they all have different body weights, you know? So to standardize things for pharmaceutical manufacturing, we usually adjust it to a standard 50-kilogram adult.
A standard 50-kilogram adult.
This gives us a consistent base for our calculations, no matter who was in the initial study, if it was mice or monkeys or a human, you know, who weighs 100 kilograms.
So it levels the playing field.
Exactly.
Okay, so we've got our starting point, our NOAEL adjusted for weight.
Right.
Now we're getting into those 5, the mysterious 5 factors.
Yeah.
F1, F2, F3, F4, F5.
F1 through F5.
Let's start with F1, which you said was species extrapolation.
Right.
And it can range from 2 to 12.
That's a big range.
What's the logic behind that range?
So F1 is where things get really interesting. We often use animal data to predict how humans will respond,
Right.
which makes sense. We can't be testing, super high doses on people.
Of course, yeah.
But, animals and humans, they're different. They have different biologies, different metabolisms.
Right, right.
So F1 accounts for those differences, how different species absorb, break down, and eliminate substances.
Right.
That range, 2 to 12, reflects how uncertain we are when we translate findings from, say, a rat study to humans.
So a higher F1 means more uncertainty.
Exactly. We're essentially making an educated guess across species.
It's an educated guess, but a necessary one.
And absolutely.
Okay, moving on to F2. This one's set at a fixed 10 for individual variability.
10, right.
Why 10? Why not 5 or 15?
Well, a factor of 10 for F2 is pretty standard in toxicology.
It's like the magic number.
It kind of is, and there's a good reason for it. We know that even within humans, there's a lot of variation.
Yeah, we're all a little bit different.
Exactly. Our genes, our health, our lifestyles, all of that influences how sensitive we are to different substances.
Right.
So this tenfold factor, it's a safety net to protect even those who might be more sensitive.
It's like a buffer for our unique biology. Okay, so that's F2, individual variability.
Right.
Now we've got F3, another range from 1 to 10. This time it's for study duration.
Study duration.
What does the length of a research study have to do with safety?
Well, think about it this way. If the studies that helped us determine that initial NOAAL were shorter than the potential time someone might be exposed,
I see.
we don't know as much about long-term effects.
Right.
So F3 increases. You could go all the way up to 10 when the study is short. It's like adding a bigger safety net because we don't have all the long-term data.
Longer studies, smaller F3. Makes sense.
Exactly. It shows how important those thorough investigations are.
Yeah, for sure. All right. Next up, F4, severe toxicity. Another 1 to 10 range.
Severe toxicity.
When do we need to consider this factor?
So F4 is kind of reserved for those substances that have the potential to cause some serious, irreversible damage.
Like the really bad stuff.
Yeah, like cancer or damage to your genes or even your nervous system. If we see that kind of severe toxicity in the studies, then a factor between 1 and 10 might be used to further lower the permissible exposure. It's a reflection of that heightened risk.
Oh, so like an extra layer of caution for the really dangerous substances.
Exactly.
Okay, last but not least, F5.
F5.
Variable factor. And this comes in when a no effect level hasn't been established.
Right.
So what happens when we don't have a clear no effect level?
Well, sometimes the data we have just doesn't give us enough confidence to set a precise NOAAL.
It's like we're missing a piece of the puzzle.
In those cases, we want to be extra careful, right?
Of course.
So we use F5, a variable factor, usually bigger than 1, to lower the acceptable exposure even further.
It's like we're playing it safe until we get more data.
Exactly. It's a precautionary approach, acknowledging that we don't know everything.
I like that. It's a humble approach. So when we take a step back and look at this whole formula, it's like all these F factors, they're like brakes on a car.
Yeah, I see what you mean.
They're progressively making that daily exposure limit more and more conservative.
It's all about minimizing risk.
Right. So we start with NOAL, our basic understanding of safety, adjust for weight to make sure we're comparing apples to apples.
Leveling the playing field.
Exactly. And then each of these F factors comes in, adding a layer of protection, accounting for all those unknowns and potential risks.
You got it. It's a multi-layered approach that really shows how cautious the pharmaceutical manufacturing process is.
It's reassuring to know that they're being so thorough.
Absolutely. It's a systematic way of dealing with the complexities of toxicology and, just the fact that everyone's a little bit different.
So thinking about all those layers of safety built into determining a permitted daily exposure.
Yeah.
What does this tell us about how seriously the pharmaceutical industry takes safety, and how could we apply these same ideas, these layered approaches, in other fields where there's potential risk?
Cleaning validation, especially in facilities making multiple products.
Right.
Using the same equipment for different drugs. Our deep dive is going to focus on getting a real grasp of maximum allowable carryover, or MS as it's called, which is central to preventing cross-contamination.
Definitely. Yeah, you've pulled some great material together for this.
Right.
That outlines a typical approach to setting these limits, and the MP value is really at the heart of it all.
Okay.
It basically defines how much leftover residue from one product can be left behind in the equipment.
Okay.
Without posing a risk to patients who take the next product made in that same equipment.
Okay, so that's the what. But what's the how? How do they come up with that specific limit? What goes into deciding that?
Well, there's a specific calculation involved, and the sources lay out the formula pretty clearly.
Okay.
It boils down to this. MAC equals the permitted daily exposure, and we can call that PDE of the previous product. We'll just call it product A here for clarity.
Okay.
Multiplied by the volume of the first batch of the next product B.
Okay.
All of that is then divided by the maximum dose of that second product B, and the surface area of the equipment they both came into contact with.
Okay. So that's kind of like the nuts and bolts, right?
That's the core of it.
Yeah, the core of it. But I think, for our listeners, it would be really helpful to kind of unpack each of those pieces, starting with the first one you mentioned, PDE, permitted daily exposure for product A. What does that actually tell us?
Mm-hmm. Well, think of PDE as like a scientifically determined safety level.
Okay.
It represents the amount of a particular substance that a person can be exposed to every day over a lifetime.
Okay.
Without any expectation of harmful effects. And these limits are not just arbitrary. They are set by toxicologists based on extensive research.
Oh, wow.
And often they actually build in extra safety factors just to account for any unknowns or variability.
Okay, so that's like the baseline for that first product.
Yeah.
And then, okay, moving on. The next piece of this formula was volume of the first batch of product B. Why is that volume important? Why does that factor in?
Well, because that represents the immediate potential for exposure to that leftover residue.
Okay.
Even a minuscule amount of product A leftover could spread throughout that entire first batch of product B.
Okay.
So by using the minimum batch size in the calculation, it creates like a conservative safety margin.
Oh, I see. So you're really planning for that.
You're accounting for
the smallest amount.
The smallest amount
of that new product that could be affected.
Exactly.
Okay, that makes a lot of sense.
Mm-hmm.
And then the next part of the formula is the maximum dose of product B. How does that play a role?
Well, that takes into account how much of the new medicine a patient will typically be taking, right?
Right.
So if you have a higher dose of product B, that means that even a tiny amount of carryover from product A could result in a more significant exposure for the patient.
So it's not just the amount, it's the concentration. Right?
It's the ultimate concentration that the patient receives.
That the patient receives. That makes sense.
Yeah.
And then finally, surface area of shared equipment.
Yes.
So that's like the part of the machinery that both of those products actually come into contact with.
Precisely. And this is the area where residues from product A could potentially linger.
Okay.
Even after cleaning. Right.
Mm-hmm.
So a larger surface area obviously means there's more potential for carryover into the next product.
Right. It's like, imagine trying to clean a huge, like a really intricate piece of equipment. There's just so many nooks and crannies.
More places for things to hide.
Exactly.
Yeah.
So by kind of taking all those pieces into account, the safe exposure level of the first drug, how much of the next drug is being made, the dosage of that, and the size and the shape and all the intricacies of that equipment, you arrive at this MAC value.
Precisely. And that is the maximum allowable amount of the first drug that can be present in the second drug.
And that's based on science.
It's all grounded in science, and it gives us a way to define what a safe level of cleanliness actually is, quantitatively.
So it's not just like, oh, it looks clean.
It's not subjective.
It's an actual number.
It's a measurable, science-driven limit.
Wow.
Yeah.
And then this MSC number, this is what is used to actually validate cleaning procedures.
Exactly.
So if a company can consistently prove that they are cleaning the equipment to a level below that MSC, they've basically proven that their process is safe.
That's the fundamental principle,
Yeah.
and it provides that health-based criteria for ensuring drug safety in these multi-product facilities.
It's amazing how like something that seems so complex can be broken down into this like logical step-by-step calculation.
Yeah.
I think the big takeaway here is that amicus isn't just some random number. It's a science-driven limit designed to protect patients from potential cross-contamination.
Yeah, it really makes you think about how this kind of approach, this risk-based quantitative approach, could be applied in other industries as well.
Oh, absolutely.
Where even trace amounts of contamination could be a huge issue.
Yeah, it makes you think about like food production or
Absolutely.
cosmetics or
Anything where purity is paramount.
it's all about understanding the risks and then figuring out those limits
Yes.
and setting those clear scientifically backed boundaries.
And having those measurable thresholds.
Right, to manage those risks effectively.
That's it.
That's so interesting.
Yeah. Stage 3 of the cleaning validation lifecycle. And honestly, just saying that out loud makes it sound a little dry, but trust me, this is crucial stuff.
It really is, because this is where we prove that the initial cleaning validation isn't just a one-time thing, but something